Hello and welcome back to the Science Explorer. If you haven't joined us before, my name is Tiffany, and if you have, like I say, welcome back. This is a technology and innovation special that is kindly brought to you by Molecular Devices. Now, Molecular Devices have provided their customers with innovative bioanalytical solutions for over 30 years, and they help advance discovery in protein and cell biology around the globe. Today, I am really happy to be joined by Justin Dranschak, who will be discussing how Molecular Devices are supporting scientists that are researching COVID-19 cellular responses as well as vaccine development. You can also find out more about the company by clicking the link in the description below. But without further ado, thank you for joining us today, Justin. It would be really great if you could just provide a brief overview, introduce yourself and your role at Molecular Devices.
Yeah, thank you very much for the time. I appreciate it. Yes, I'm Justin Dranschak. I'm the North American manager for Molecular Devices BioPharma portfolio. Been with Molecular Devices for around 9 years in various roles supporting our customers through bioresearch, high content screening and imaging, and now through our biopharma platforms.
Perfect. So, I mean, we're obviously in a precarious time at the moment. So, what are the current concerns and needs that scientists have during this pandemic, and how are companies like Molecular Devices managing to fulfill those needs?
Yeah, I think that's a great question. I think the initial concern for the scientific community in general is really, to get a complete understanding of how this virus and disease kind of works. And, it presents itself through various symptoms and interactions from patient to patient. And so there's really a high demand for accelerated testing and medicines and being able to identify how to get those to market as soon as possible and to kind of fast track discoveries, either based off of current and existing research or through new avenues of research that might have presented through the broader scientific community globally. And so that's really kind of where Molecular Devices and companies like Molecular Devices are stepping in to support the research community through providing different technologies that help them accelerate those solutions and really optimize their workflows to take something from a lead target or molecule all the way through that discovery and analytical testing process to a finished product that results in a therapeutic or a vaccine or a solution to help combat the current situation that we're in.
Perfect. No, that makes sense. So, can you tell us a little bit more about how the products fit into this kind of COVID-19 research environment?
Yeah, so our customers really are focused in a wide range of different applications across kind of biological research. And given the current COVID environment, a lot of our customers have pivoted into vaccine-based research, focusing on recombinant proteins and monoclonal antibodies, and then also repurposing existing drugs that have already been approved or are in clinical trials and helping to develop those, into potential vaccines. And so Molecular Devices platforms really fit across all aspects of that development process. So we have platforms that are upstream, kind of in the build and design phase of an experiment, and workflow down through the actual testing and characterization of lead molecules and targets, all the way through to where they would actually take that into scale up. And so we deal with high content clonal screening and selection, allowing researchers to identify cells that are producing a high amount of a specific antibody or protein of interest. From there, they can then, characterize those and look at how those interact at a cellular level, both internally using our high content imaging solutions. And then even further from there, look at different applications in terms of viral efficacy and titer and how those actually work against or with cells in certain ways at a microplate level. And so we have microplate detection technology that utilizes different types of analysis like absorbance, fluorescence, and luminescence characterization, providing researchers a much better understanding of what their end goal really is for their molecules.
Mm-hmm. That's fantastic. I mean, I knew it was broad, but I didn't quite appreciate how broad we're talking.
Exactly. It's a wide range of different analytical platforms that really come together across multiple workflows.
Yeah, no, that's phenomenal. That really is. So, I mean, we touched a little bit on it, but considering immunology then, so can you explain a bit more about this area and then give us some examples of some more workflows that are being used in immunology research labs at the moment?
Yeah, sure. So it's really kind of broken down into 4 different areas for where customers might be focusing. So in immunology, there's a basic research focus where you're looking at more of the genomic profiling of the disease and really what it's made up of, right? Then you're going to look at kind of the mechanism of action and the different viral pathways, how it infects people. And from there, once you have that kind of basic understanding, so those were kind of the initial things done by the research community, involved with COVID. And then you really move into more of candidate vaccine, candidate therapeutics, and diagnostics. And so for candidate vaccines, you're looking, again, at kind of viral efficacy, how it infects, how much, how it interacts, and really trying to create a target profile for your molecules. And then for candidate therapeutics, that would be where you're looking at different types of immunotherapies, ways to combat that might not be a vaccine, but that might inhibit the virus or inhibit different types of disease models that affect humans, and being able to repurpose some different things that way. And then in terms of diagnostic, that really falls into where everybody is interested in right now in terms of being able to detect, right? If you have COVID, having those tests to detect for those types of antibodies and be able to, look at, how can we determine, how much antibodies someone has, whether they've, been infected or come into contact with that. So how does molecular devices kind of fit into those areas? We really incorporate our instruments and our platforms into a wide range of different workflows. And so for us, probably the biggest one is going to be in immunogen and antigen discovery. And this is where, it's a process for identifying, a protein or antigen associated with a specific immune response. And the way we do that is our platforms, a lot of the times, the only way to identify specific proteins or immunogens of interest is to use large bacterial libraries that display genetic information on their surface. And so we have high throughput microbial colony pickers that were actually used in the Human Genome Project for screening thousands and sometimes even millions of different variants, and allowing researchers to quickly identify based on size, shape, morphology, different characteristics, fluorescence expression, to identify the clones and the colonies of interest that are displaying that information that they need. And then they take those and transfect them into mammalian cells. And from there, then begin to screen with like our high content imagers or our plate readers for quality attributes such as viral titers or efficacy or viral infection, all of the different types of characterization applications that they might be working on. It's also known as phage display workflows as well. And then we've got more directly on the mammalian side that deals with antibody discovery and more in the hybridoma screening. And so to start with those workflows, Molecular Devices really has high throughput colony screening platforms on the mammalian side that allow you to screen tens of thousands of colonies and clones very quickly and identify maybe that top 1%, top 2% of clones that are producing the most of your specific antibody of interest. And our instruments will then pick those out of wells that they're growing in and then deposit those colonies into 96 well plates for further downstream analysis where they'll grow up. And then they'll begin those characterization screens, looking at how those, how those antibodies, how they can, harvest those antibodies to look at how they, block viruses or interact within human cells. And so those are probably two of the major workflows, but also within cell line development and gene editing as well. we play a predominant role for CRISPR-based gene editing
Okay.
experiments where labs are looking to knock in or knock out a specific gene of interest. And Molecular Devices offers higher throughput solutions that allow labs to start from a single cell. So one of the requirements for immunotherapeutic or biologic development in terms of monoclonal antibodies or other things is that researchers, one of the main bottlenecks is you have to prove that your therapeutic came and started from a single cell,
Right.
so that you don't have any variability or anything like that. And so Molecular Devices have been focused in this area for years and years. And right now we have a really high throughput workflow that allows you to deposit single cells using a single cell printer, or Clone Select single cell printer, and it'll deposit a single cell into a well and provide image-based evidence of that sorting event so that you have confirmation that you are sorting one cell of interest. Following that up, we have a high throughput single cell imager so that you have documentation post-sort, that you have one cell, and then you can track that and the growth characteristics associated with it as it grows from a single cell into a colony. And so from there, you're normally going to do additional types of screening on other instruments like our plate readers or high content imagers to look at how those cells interact and how your gene of interest may interact, if you're looking at different types of proteins or immunogens or things like that, which would then further take that downstream.
Wow.
It's
a lot to process, isn't it? It's phenomenal. Wow. No, it's great. I mean, so I wanted to kind of talk about what you're doing to help your customers streamline your workflows. And then obviously with that will come, accelerated time to results. But I guess that goes hand in hand with potentially discussing any challenges or limitations that maybe either you face or your customers have faced that have then allowed you to kind of work to streamline, if that makes sense.
Yeah, that completely makes sense. And really, I think the biggest hurdles for our customers right now with these workflows is the full-time employees to do the jobs, right?
Yeah.
I think everyone is, sheltering in place and in various different positions where they might have access to their labs, they might not have access to their labs. And so that's really where Molecular Devices has been able to help. So we have automated workflow solutions groups. And so labs that might have the funding from either the government or their institutions or companies to do the research, they might not have the means or the manpower to kind of implement that. And that's really where our instruments and our platforms help provide them a solution because it is higher throughput. So they're able to do more sample testing at greater speeds. They're able to screen more cells faster on our colony picking platforms. They're able to run more tests in 96, 384, 1536 well plates on our plate readers or our imagers. So they're allowed and able to move what they're looking through, their process, much quicker to determine which direction they want to go from there. But in the larger scheme of things, we are working at more automated workflows where we can integrate multiple instruments of ours into one seamless workflow with robotic integration, where we will actually take plates from one instrument into an incubator where cells will grow. We will take that out with another robot and put it onto an imager where we'll image, and then it'll go back to an incubator. So we can automate the process of multiple different people into one uniform solution that allows them to get from their starting question and analysis through to that end product much faster.
Perfect. No, that makes sense. Thanks for clarifying that. So I guess, so what kinds of customers are currently using these workflows then and your instruments? And then who else do you think should consider using the expertise of your company?
Yeah, that's a good question. So right now, our customers that are using these solutions really range from basic academic labs all the way through government institutions, federal government institutions, state, local, up through small, medium, and large pharma and biotech. So we really run the gambit of kind of all biological research labs and institutions. But now I think more than ever, we're branching out into labs looking at doing single cell sequencing and RNA sequencing with our single cell printers. A lot into antibody discovery and protein engineering, but then we also work with food and beverage and really agricultural sciences as well, in terms of looking at genetic modifications to crops or whatever it might be, and how those might affect certain products in different industries that way as well. So there's a wide range of different kind of applications on it. But right now, I think obviously most of the focus has been in the immunological area, and how labs, even who are just doing basic drug discovery, can kind of pivot into the vaccine and viral-based research applications.
Perfect. I mean, definitely a force to be reckoned with is what I've got as my concluding remark. Well, I mean, thanks so much. I think we really appreciate you taking the time out to explain this and go through all of this in so much detail. I hope everyone at home has enjoyed this. And if you do have any questions, please just leave them in the comments, and we'll make sure that we get to them as soon as we can. Thank you again, Justin.
Thank
It's
you.
been great speaking to you.
I appreciate it. Thank you very much.